ಸರ್ ನಮಸ್ಕಾರ.
ಇಲ್ಲ ಸರ್ ಬಂದರೆ ಈಗ ಆ್ಯಕ್ಚುಲಿ ಸರ್ ಟ್ರಾವೆಲ್ ಮಾಡ್ತಿದ್ದಾರೆ. ಹಾಂ 3:30 ಇವರು ಡಿ ಸರ್ ಅವರೇ ಲಾಗಿನ್ ಆಗಿರೋದು. ಫಿನಿಶ್ 3:30 ಜಾಯ್ನ್ ಆಗಿ ಲಾಗಿನ್ ಆಗಿ ಬೆಟರ್ ಅಂತೀನಿ ಸರ್. ಲಾಗಿನ್ ಆಗಿದ್ರಿ. ಸೊ ಅದೇ ಸರ್ ಈಗ ಸರ್ ನಡ್ತಾರೆ ಸರ್. ಅದಕ್ಕೆ ಶಿರ್ಸಿ ಹಾಂ ನಿಮ್ಗೆ ಗೊತ್ತಾಯ್ತಾ? ಅದಕ್ಕೆ ಬೇಗ ಮುಗಿಸೋಣ ಅಂತ ಗೊತ್ತಿಲ್ಲ ಸರ್. ಲಾಗಿನ್ ಆಗಿತ್ತು, ಅದು ಕಾಣಿಸ್ತಿದೆ ಎಲ್ಲೇ 3:30 ನಾ ಕಾಲ್ ಮಾಡ್ತೀನಿ. ಇಲ್ಲಿ 3:30 ಲಾಗಿನ್ ಆಗಿದ್ರೆ ಬೆಟರ್. ಇಲ್ಲ ಸರ್ ಮಾಡ್ತೀನಿ ನಂದು ನೀವು 3:30 ಲಾಗಿನ್ ಆಗಿದೆ ಸರ್. 3:30 ನಿಮ್ಮದು ಮೇನ್ ಇರೋದ್ರಲ್ಲಿ ಕೋಟಿಕಲ್ಚರ್ ಒಂದಿಷ್ಟು ಮಾತಾಡ್ತಾರೆ. ಪ್ರೆಸಿಡೆನ್ಷಿಯಲ್ ಮಾಡ್ತೀನಿ. ನಿಮ್ಮದು ಮೇನ್ ಸರ್ ನಮಸ್ಕಾರ ಸರ್. ರೀಚ್ ಆದ್ರೆ ಸರ್ ರೀಚ್ ಆದ್ರೆ ಸರ್. ಹಾಂ ಸರ್ ಜಾಯ್ನ್ ಆಗಿ ವ್ಯಾಲಿಡಿಕ್ಟರಿ ಅದೇ ಪ್ರೆಸೆಂಟೇಷನ್ ಸರ್ ಒಂದು ಮಾಡಲ ಸರ್. ಓಕೆ ಥ್ಯಾಂಕ್ ಯು. ನಿಮ್ಗೆ ಫೋನ್ ಮಾಡ್ತೀನಿ. ನೀವು ಲಾಗಿನ್ ಆಗಿತ್ತು ಸರ್. ಈಗ ನೀವು ಲಾಗ್ಔಟ್ ಆಗಬೇಡ್ರಿ ಸರ್. ಯಾಕಂದ್ರೆ ಲಾಗಿನ್ ಆಗಿ ಯಾಕಂದ್ರೆ ಆಮೇಲೆ ಅವ್ರು ಬಂದ ತಕ್ಷಣ ಬಿಡೋದೇ ಇಲ್ಲ ಸರ್ ದಾರಿ. ಪ್ಲೀಸ್ You are presenting it. Girish. Hello.
Hello sir.
Girish, voice ಕೇಳಿಸ್ತಿದ್ಯಾ?
ಕೇಳಿಸ್ತಿದೆ ಸರ್.
Okay, thank you.
Okay.
Good afternoon, everybody. So today's session we actually had two different sessions, but due to the want of time, what we have decided is that we'll take up only one. So the valedictory ceremony we try to finish it off in 3:30. 3:30 we'll commence at 3:30 today, and we'll try to finish it off in half an hour, 1 hour, something like that. That's why we have decided to take up only one presentation today. And the presentation which I'm going to do today is the recent advances in insect pest interaction for the insect pest management. So this is the topic that I'm going to make the presentation on. I will not go into details as to why this integrated pest management is has to be relooked, because we have talked quite a bit about the various aspects of it. Okay. So So we'll not go into detail as to why that we have to look at the integrated pest management strategy again, because the reason for the quest into the integrated pest management started with the deleterious effects of the chemical pesticides that we have actually had in the early part of the 60s and 70s, and that's the reason that why we decided to go for the integrated pest management strategies. But the fact of the matter is such that, like, we integrate all the components which we have decided to put in the integrated pest management. They were actually the arbitrary in nature, and there was no scientific base. And once we started relooking into the integrated pest management strategies, we had to look at the various aspects, various dimensions that are involved in the designing the IPM modules. And apart from that, the IPM models which were put into the use on the community basis, they were not finding the momentum. So because of the technical stack of the technical base which was wanting into that, so we also have the on one hand the integrated pest management strategies, and the other thing is that we also have to have alternative approaches. And if we don't do that, this is very imperative, because reducing the risks and the impacts of pesticides. So pesticides used on the human health and the environment, that's one thing. And on the other side, promoting the IPM and use opportunities. So that has to be looked into the system. Otherwise, this is the place where we are heading towards, like, they will not be able to strike the balance in the food production and the control of the insect pests, and they will be falling apart in combating the insects. That's why there is there is a lot of emergency in finding out the alternatives for the integrated pest management strategies. And apart from that, so what are the various aspects that are involved in the insect-plant interaction? So we have the slogan of the day, which is like, more crop per drop, and for that matter, we will have to look into the insect-plant interactions to find out the intricacies that are involved in the interaction between the different trophic levels, which is highly complicated and dynamic as well. So this is exactly the system has worked till now, since the prehistoric period. Different timeless associated with the insect-plant interactions is that there has been a co-evolution of the insects in the plants for over the 400 million years, and it does not take much time for the insects to alight on the plants, because it will take only a few milliseconds for it to alight on the plant surface. And see, once it alights, what it does is, like, it will colonize itself in a few minutes, and once it colonizes and starts causing the damage, what the plant exactly does is that, like, it will start defending itself. So for every offense, there is a defense from the plant system, and this is not as simple as that, but otherwise the impact is very complicated. That's why we'll try to see what I had is a lot of information that I had gathered, but what I've decided, like, we'll not go into details of all these aspects, because one of our speaker, in fact, while speaking about the semi-chemicals, he has elaborated. So whatever the top work that has been done, so the practical level, so I try to cover that one. So this is exactly the works. So the first trophic level is the plant. This is what I was talking about. And the second trophic level, the food level, is the pest which is going to affect the plant or infect the plant. And this is the third trophic level, which is the third trophic level, which is the natural enemy, of course, like, which is going to affect the pest which is going to cause damage on the plants. So the first trophic level, what exactly it does the plant is, like release certain infochemicals, so which were received by the pest, which is an herbivore, and that in turn will get, it will elicit a sort of response from the pest, and the pest again will release certain chemicals which are released by, they are called as the pheromones, which has been covered already in the previous topic, the previous speaker. So what I'll do is that I'll not touch upon all those things. So these pheromones are released to the environment, the atmosphere. So they are actually received by the natural enemies, and this will elicit a specific response towards the pest, and the infochemicals that are released by the plant, again, they are received by the natural enemies, and this will give an invitation. This will give an invitation to the natural enemies towards the host plant. That's how the tritrophic interaction takes place. And I will not go into the details, but this is the basic funda which is involved in the insect plant interaction. It is only at the 3 trophic levels. So there are the arm race, because for every offense, which is an evolutionary product, there is a defense by the plant. For potential creation, there is always a potential destruction which is counterbalanced, consequent to which there will be the balance of life. So as and when the pest evolves itself to become the new offense to consume the plant, so the plant will develop its own defense to counter the attack by the insect, and that's how the evolution of the defense takes place. And this is not in one go or one night it will take place, because it is generally said like Rome is built not in a day. It has taken several thousand years, and similar way the defense of the plant and the offense of the insect is not developed in a day, rather it is a product of the evolution. So this is exactly, for example, so the insect, for example, like the plant, if it develops a certain toxin, so to counter the insect, if the insect attacks and then it releases the one toxin, to which there is a toxin which is produced by the plant system again to counter the insect attack, and if the second toxin is released by the insect to cause infection on the plant surface, so there is a second toxin again which is produced by the plant to counter the insect attack, and likewise it will go in sequence or succession. So that's how the potential creation is counterbalanced by the potential destruction. So potential creation here is with the plant per se, and the potential destruction we are talking about the infection or the infestation which is caused by the insects. And I'll not go into the details of all these things like in the armed diversity of the plant defenses. So one way, of course, line of defense the plant exactly which follows is like avoidance. So what it does is like we try to escape from the very susceptible stage. That's what we call it as a avoidance. And the resistance is also one second the prong of defense, wherein the plant will try to reduce the fitness of insects after the contact. So this may either be in terms of the preference, non-preference, so which is purely the morphological in shape, and otherwise there will be anti-biases also, which is afforded by in fact the alkaloids which are synthesized in the plant system, biochemical system. And the third defense, in fact it is not the defense, what I would say is like another host plant resistance mechanism, so wherein the insect will continue to attack plants, but the plant will withstand all the damage which is caused by the insects, and it will continue to grow in size, and in fact it will put forth the sufficient amount of the biomass. So that is called the tolerance. And at the same time, the insects, as the plants have the certain defense mechanisms, there are certain the offensive mechanisms which are actually exercised by the insects. That is called counter defense. The insect actually defends itself by the plant defense. So how exactly it does is the first and the foremost is, and the wish is the behavior of avoidance. What exactly it does is like there is a particular area, the crop plot which has been sprayed with the chemical insecticide, it will try to avoid. So the visiting that particular place. Otherwise, what it does, it will first in the first go it will stop feeding, and then if the need be, it will stop even the ovipositing or laying the eggs also. And there is a modification in its own behavior as well, the biochemistry to overcome the plant defense. The first of which kind is like the counter defense by way of the behavioral mechanism. And the second one is the biochemical mechanism. And there is also a possibility like actively actively manipulate the insects will try to actively manipulate the host plant's nutrition and defense. We'll see one by one. So the first segment of the counter defense is like the behavioral defense, wherein the insects will try to cut the veins or manipulate the rolling and mining between the epidermal layers, and the gregarious feeding by the insects when they go in the mass or congregation. That is also that they will try to intimidate the plants, and this is the behavioral offense mechanism which the insects generally follow. And the biochemical counter defense is also there in case of insects, and the first one is the rapid excretion, whereby the mediation, like the possible use of the toxicant which has been ingested is avoided. It will be quickly thrown out, and the sequestration of the toxins. So the chemicals which have been taken in, what they generally try to do is that they'll try to send it into the biochemistry, like the biochemical mode for sequestration, and there is a possibility of detoxification of the toxins also.
So once the toxins which have been they sprayed into the system, enter into the system of the insects, they get detoxified by way of enzymatic action, and the target site sensitivity is also the fourth step by which the insect's counter defense is operating. So what they do is there may be certain features which may not be palatable or which may not be the attractive to the insects, but the insects will try to develop a sort of insensitivity to the target. And insect counter defense by way of manipulating the host plants is that so either they will produce the galls or otherwise the abnormal structures wherein the gall. So for example, the aphids, wasps, and the mites, they will try to develop the sort of the galls, and they also try to manipulate the defense of the plant itself. And this is exactly the herbivory will be there, and the feeding by the insect, and the arm race for the counter defense will be there, and diversity. See, the second one is the weapons which are used by the two different trophic levels, the diversity of the plant defenses, the diversity of insect offense, the counter defense. And there is also a signaling interaction. The signals plants the received from the herbivores, and the signals insects perceived by the plants is also one way that they will try to interact with each other. So the plant insect signaling interactions generally are falling into three different categories. So they in fact are the signals that the plants receive from the herbivores and other plants to defend themselves by their wounding. So the caterpillars are in the stage of the insect which is damaging, what it does, it will cause the mechanical damages, and to which the host plant will become alert. And herbivore associated molecular patterns, which is activated as a HALPS. So there are certain chemicals and there are certain enzymes also which will detoxify, which are released immediately to detoxify. And talking trees. So suppose there is a group of plants at a plant individual, when it is attacked by the insect, what it does is that it will try to emit either a single chemical or a group of chemicals, and which on being received by other plants, they will become alert and they will try to develop their own defense mechanism against the possible danger which is likely to be caused by the insects which is onslaught. Okay? And the signals that plants receive from the herbivores is the first one is the wounding. On being wounded, either green blade or important plant, what exactly it does in certain cases that it will develop synthesize the joxaminic acid, which is a defense mechanism, which is the first line of defense in some of the plant species. Okay, and apart from that, there are salicylic acid and ethylene also, which will actually try to counter the insect pest attack. The plant insect signaling interactions, the signals that insects receive from the plants, plant defense compounds like allelochemicals and the proteinase inhibitor and the plant defense signaling hormones. These are all the counter defense mechanisms by the plants. Okay, and types of plant insect interactions, we always believe an insect is an herbivore and it is going to cause the damage to the plant, and the plant in turn will counter defend itself to make sure that there is the feeding does not continue. Feeding does not continue. But the fact of the matter is there are several other associations also. So first one is a mutualism, wherein the flowering plants and the pollinators or any other insect species, they will coexist and they are benefiting each other, and that is called as mutualism. And there is an antagonistic herbivory. This is quite this is the one which is most talked about, because from the pest management perspective, this is more important. And apart from, say, here in this case, the herbivore obviously is benefited and the plant gets affected, and the antagonistic insectivory is the third, the segment which is there in the insect-plant interaction, where the plants eat the insects for the nitrogenous the nutrients. For example, the pitcher plants that I'm talking about. The coexistence by the insects is the coexistence between the insects and the plants is such that they will coexist without being harmed and without being benefited also, and that is called the commensalism mechanism. So the insect effectors suppress or induce the plant defense. That is by way of releasing So by way, say for example, there is an insect either chewing and biting that insect. More likely it is the piercing second type mouthparts for the insects have. So what they generally do is that they probe into the system, and consequent to which there will be release of certain chemicals for the pre-digestion of the food before being taken into before being ingested. So all these the enzymes which are released into the system for the pre-digestion or the partial digestion of the food before being taken inside and assimilated. So the plant also becomes alert, and consequent to which there will be cellular signaling like reduce the immune response will be there, and affect us in saliva. They will get affected. And the difference is quite often we say it is only the tolerance, resistance, or immunity, or the preference difference. But when we look into the abstract idea or the abstract aspect of the defense is that first and foremost is the constitutive. So which is the genetic makeup of the individual which will afford the resistance against the insect, and that is called as a constitutive defense, which comes through the breeding traits of the species that I'm talking about from the plant perspective. And induced defense is such that once after being attacked, the plant starts developing certain traits, so that will elicit certain signals into the system and attacking the organism and the volatiles that get to the plant activator. These are the induced defense, so which are also only possible on being attacked by the insects. And the primed defense, and this is primed defense is very novel defense mechanism which has been discovered by the scientists. So when the plant is ready to mount a quicker and a stronger defense when subsequently attacked, because it is only because of previous experience, the plant would have developed its own resistance mechanism to counter the specific the attack or the infliction by the pest. So the host location, as we have seen in the first slide itself, the insect does not take much of the time for its feeding. So once it is in the flight on the way, it alight on the plant surface, and the conveniently closer leaves or the any part of the plant they are actually fed. So the decision making is much quicker. And how do the insects recognize the host plant? Either it is by the single volatile, or it may be sometimes the combination of the volatiles which are released by the host plant that will elicit a specific response in the insect which will receive the insect, and either that will deter or attract the insects towards that. In the sense either the positive or the negatively oriented movement will be elicited in the receiver. So quite often, all these host plant resistant mechanism of the insect plant, the interactions, they all revolve around the polyphagous pest or the most obnoxious pest, like the RHRS, because it is highly polyphagous and it specializes on the reproductive part, which is an economic part from the cultivation perspective. So attraction to the proper blend of the chemicals, unless it is there, the insects will not get attracted. If it is the proper blend is there, the insects will make the positive oriented movement towards the source. Otherwise, so they will get retracted. Otherwise, they will get repelled. They will move away from the source of from the source of stimuli. And the techniques which have been employed. So there were they had totally 123 slides to talk about. But what I personally felt is all the work that has been done. So which is basically the more chemical ecology in nature. But what I personally feel is that the top work that I've done during my PhD work, I just want to share a few experiences that I had during my PhD. So this is this is the system, the component 1, that is the IPM model 1, and the component 2, the second IPM model that we have developed, and this one, the third one, is the recommended package of practices, which will be the chemicals which are as per the recommendations, the cotton. This is the cotton crop that I'm talking about. This experiment was done in the cotton crops. So the package of practice is whatever that is mentioned, oxalimetol methyl, the monocrotophos, quinalphos, sulfamethrin, chlorpyrifos. These are all the schedule of this is the schedule of the chemicals which is there in the package practices. And the standard IPM package or integrated pest management module, which is generally followed, this is the first one that I'm talking about. And I didn't do anything more. So what I've done is that I have made a slight variation in this one. At the 50th day and the 57th day, the Jaidar leaf extract, the spray which is preceded before the release of the Trichogramma chilonis, so which was to the tune of 2.5 lakhs per hectare. And again at the 57th day, so before releasing the Trichogramma chilonis, so the Jaidar leaf extract was sprayed, and it has given a more enduring result. So there is a scientific evidence that why I did that. It was not done arbitrarily. I'll tell you why I did that. So attraction, so the basic work which I did in the infochemical mediated interaction or the post-plant reaction, post-pest reaction, post-pest and even the third trophic level, the Trichogramma chilonis. So which I started with the small experiment. So wherein the attraction of the Trichogramma chilonis to the post-plant, that is the post insect, that is the Helcor parmigera, with and without a host. The Jaidar cotton was taken as the host. So Trichogramma chilonis exposed to the Helcor parmigera eggs on the cotton leaf and on the filter paper. There is significant variation in the number of encounters that by using the antenna. So the kind of encounters the Trichogramma chilonis which it did was far higher in case of the Trichogramma chilonis when it was offered with the Helcor parmigera eggs on the cotton leaf compared to the filter paper. It was almost 50% more. So on the encounters made in case of the Helcor parmigera eggs when they were offered to the Trichogramma on the cotton leaf blade. So that is the impact of the post plant that's what I'm trying to tell you. And the second one is the influence of the substrata on the parasitic competence of the Trichogramma species. So when anyway we have come to know there is a significant impact of the host plant when the significant impact of the host plant on the eliciting the specific response to the Trichogramma. But which particular part is more competent in eliciting a response? This I have done the experiment, and the observations were taken 3 and 5 days later. So what I could see is that the amount of parasitism which was seen on the leaf blade, which by all the species, in fact, like Trichogramma pretulosum, Trichogramma japonicum, Brasiliensis, and chilonis, it was all higher when the Helcor parmigera eggs. So they were offered to the Trichogramma chilonis on the leaf blade compared to the poles. And this is the influence of the substrata for the parasitic competence of the Trichogramma species. So this is the 5th day observation. I will not go into the detail like Polakian. So this is the diagrammatic or the pictorial representation of a response which was seen in the 3 days after the release and 5 days after the release in the Trichogramma species. And this is corollary, if the percentage of the percent of parasitism is basically the hatching will be the much lesser. So this is seen in the percent hatching. So this is the So what I did for my experiment is that I have taken the 4 different species of the cotton: Gossypium hirsutum, Gossypium arboreum, Gossypium herbaceum, and the Gossypium barbadens. One cultivar, each one of them representing the species, like DR6 in the hirsutum, and AK235 in case of the arboreum, and Jaidar in case of herbaceum, the cotton species, and P82-11, so from the herbaceum, because all of them they have the different plant architecture altogether, especially the trichomes so which will either attract or repel, or it will make a specific the response in case of the Trichogramma species. So what I did is that the influence of the cotton species on the parasitization of the Helcor parmigera eggs by the Trichogramma species was significantly varying. When you look at the Jaidar, which is basically herbaceum, you just see it. So the herbaceum species, the percent parasitism by all the species was much higher in case of the Trichogramma chilonis, and it was slowly decreasing as we went towards the hirsutum species. And this is the diagrammatic representation, and the percent hatching. So this is exactly the corollary the reciprocal to the percent hatching. And this was in fact proved even by the attraction of the Trichogramma chilonis to the different bud washes of the different cotton genotypes. So again the Jaidar, so it attracted 5.7, which is the highest number of the attraction by the different among the different species among the different cultivars belonging to the different species of the cotton. And this experiment was done by using the 8-arm olfactometer. And this is what I had used for the olfactometer study, which is the 8-arm olfactometer. So into the material, so I'll not go into the details. Only thing is that mated pair of, so the 8 mated pairs will be released here, and on all the sides, like different the bud washes, which are the hexane extracts, the cinnamons extracted in the hexane, they are put here, and it will give the trichogramma inside will be given the free choice, so to get attracted towards any of the preferred the cinnamon. So when we tested further, like the effect of the plant parts on the orientation of the trichogramma colonies, so the number of the trichogrammas, the pairs and the trichogramma individuals rather, so they got attracted much higher towards the cotton leaf, and compared to the bole wash and the flower wash. And in terms of the cotton genotypes and the parasitism by the trichogramma species again, so to the different species within the herbaceum like Nabadita, Sahana, Tiny Bowl, DR6, Lakshmi and the THS11. And again the trichogramma colonies, when we looked at colonies and the Brazilian species, they were on par with each other when we started. So when the finer details were looked at, trichogramma colonies was much the agile or more active in nature. And this is the diagrammatic representation. So this is the hatching, and this is the reason that why the trichogramma colonies or the trichogramma Genpachia, so trichogramma here, they are both species they have a differential, the oriented movement towards the post plant, because all the species are all the cotton cultivars which were used, they had the different plant architecture altogether. And look at the number of trichomes on the Abadita. It was the more of the glabrous in nature. And when you go further, the Sahana is more hirsute in nature. Tiny Ball is hirsute, and the DR6 is hirsute, and Lakshmi is again glabrous. And we look at the color pattern on the plant also, it is varying, the shades are different, the first go. So this is the light green, and Sahana is greenish in color, and red pigmentation is there in the tiny pole. And again, when you look at the height, so that's the overall architecture of the plant. They are again varying, and that's why the attraction also, the attraction towards the also varies. Because it is a natural instinct or instinctive preference by the trichogramma species, and this is the percent parasitism which we could see. So with the advancing post age, and trichogramma brasiliensis, trichogramma japonica, and the colonies, when we try to when we try to observe the parasitism, it was always increasing from with the advancing age, in the 77th day, highest the degree of parasitism, the percent parasitism could be observed at 77th day. So this is a clear-cut index, how exactly the host plant can influence in bringing about the specific response in the parasitoids or the natural enemies. So this is the pictorial representation of the pictorial representation of the response that is seen in the different species. And this is the percentage action and effect of pre-exposure. So we had the primary response which we saw in in one of the slides. So this is almost correlation to that one. This is an extended knowledge of the same thing. In the insects, the trichogramma colonies and the pteridosum, when they have already got exposed to the pheromones, so the fine strength on the host hex it is much higher compared to the non-exposed trichogramma species. And the behavioral response of the trichogramma species towards the cross mixture also. So when the cross mixture was given, so cross mixture, which is again the hexane extract, so the highest amount of the adults got attracted. So this is the chiramon with the scale extract. I will not go into the details of all these things, the fine spent by the trichogramma colonies and subsequently discovered eggs. And this is what I was trying to say, the second module, which was wherein before the release of the trichogramma at the 67th and 77th day, we had actually taken up the Jaidar cotton leaf extract on the Lakshmi cotton variety, and we could see the amount of parasitism which was substantially high, significantly higher, that was 27.42. So what I'm trying to say all in this case is this is the parasitism which is brought about by the trichogramma species in the Jaidar cotton variety during the year 2002. So what I'm trying to say is like, apart from the physical aspect, which is the morphological aspect, like, this is basically a conflicting demand for the breeder, because when you look at the insect-plant interaction, the different species prefers a different the type of the morphological features. For example, the Helicoor parmigera, it always prefers the hirsute type of varieties because it can oviposit easily on the surface. So the hirsute or the pubescence on the surface of the leaf blade, it gives a anchorage to the oviposition. But on the contrary, when you look at the whiteflies, so they generally prefer the glabrous surface, because so when the whiteflies or the jacies, when they alight on the surface, they make the lateral movement or the translocated movement. So because of which it will get affected by the pubescence. That's why they prefer the glabrous surface or the non-hairy surface. So what I'm trying to say is like, when you practically get involved for advocating the insect plant interactions or the products of this one. Yesterday there was a question by one of my colleagues, like Gangadhar Narvanchi. So what is asked, like, have you ever done any experiment by using the exact product which is right there in the field, which is available for the product, which is available for the farmer, which is nothing but the cool leaf extract, because farmers may not be in a position to take out these cinnamones through the hexane extractor by any other means. So you can study the plant system itself, and in fact yesterday our professor Venkatesh sir also, like, he said, like, push pull technology, and this work which I had done long time back still stands valid. That's why I decided, like, instead of taking the examples of other experiments elsewhere in any part of the world, what I decided is, like, whatever that I have done. So this is, of course, like, I have not gone into the details of the minor, the biochemical reactions or the biochemical ecological interactions at the infochemical level. So this is practically whatever that was that could be done at the farmers level that I've done. So if there are any queries in this regard, please let me know if there are any questions from anybody.
Sir, excellent presentation, sir. Many compliments first. I have one small question. Am I audible?
Yeah, of course. Arish, yeah, you can please.
Yeah. Actually, in response to insect herbivory, plants do produce some sort of defense and in the form of some secondary metabolites, right, sir?
Yes, that is true.
Hello. Yeah, my question is, suppose, without the herbivory, in anticipation of herbivory, can we do some sort of sprays by using salicylic acid or jasmonates or whatever to avoid the insect attack? And is it really practically going to work?
Practically it is going to work, I'll tell you. So what, see, the main funda, whatever that we have seen here is, like, so instead of going for the chemicals which are available in the market, so the most suitable one, like, there is the insects generally have the preference, the degree of preference for different genotypes, the different cultivars. So as it's like, you can go for the jasmonic acid and the salicylic acid, that can also be done. But whether it is suitable or not, that is under the question. But for example, if you take the cotton itself, like Jaidar cotton leaf extract, so the infochemicals which are released by the Jaidar cotton, like, they are more attractive. So this was sprayed on the Lakshmi cotton genotype. So they got attracted in search of the infochemicals which are actually from the Jaidar leaf extract. So of course you can do, but only these things, like the salicylic acid and the jasmonic acid, they are all in the experimental stage.
Okay, sir. Thank you. Because already some people have started using products like chitosan. They are already used by farmers.
They are being used, but the only thing is that they are still in the experimental stage. So we are again, like, skeptical, like, how many insects, how they will attract the insects, to what extent they will attract, and what will be the impact. They are all the things which are still to be studied. That's why, like, the integrated pest management is continuously evolving. So the various aspects, the intricacies are being studied still. So yesterday we talked about, our professor talked about the push pull technology, which is nothing but the push, and from the other side there is a pull, and such things. So it's always a continuous evolution the integrated pest management is going to go through.
Or rather, are we not misleading either the plant or insect by intervening? by taking such interventions? Whether plants or insects will discriminate this kind of interventions?
This is possible, subject to the condition that they get used to that one. So in the because the age of the human being on the earth is only 5 lakh years, and that of the insects is 350 million, over 350 million years, and naturally, so it may be any brilliant, but we cannot cheat the insects, and they in turn get used to that. That's what in the third or the fourth slide I was talking about, the behavioral, biochemical. You do any sort of thing. If you I don't know whether you had the opportunity to visit the Gulbarga or not. So in the southern part of Karnataka, so you will recommend the insecticides 1 ml, 2 ml, 3 ml. If you come to the Gulbarga area, so you recommend them the 2 ml, they will spray the 20 to 40 ml. So for every sort of action, there was a reaction by the Helicobacter parmigera. Once they decided that we will not take up any sort of the plant protection measures on the red gram, so that was like a natural response. The Helicobacter parmigera started receding. So that's why, like in the beginning, we had the concept of insect pest control. We were on onslaught to control or eradicate the entire species, which is neither advisable nor possible. We can cheat them only it is momentary. That's all.
Okay. Thank you for your inputs. And one more question, sir. Can we use colors of different lights in pest management? Of course, we know only about this. Sticky traps and other kind of coloring materials. So any thoughts on that in that line, sir? Using colored lights for deterring or attracting something like that.
Yeah, colored lights, of course. The western part of the world, there is a lot of work on that. But the only thing is that they are used only for the monitoring. Because if you imagine, like the colored traps are, the colored light traps, if they are to be used in the Indian condition, so we have to work out the economics also. And how many is the point sources of the electricity that provide? They are all the practical feasibility also we have to look at, Harish sir.
Yes. Why I ask this question is, for my PhD work on turmeric, I was using different colored nets for growing turmeric. So surprisingly, in yellow colored net, I got least infestation of shoot borer. I am finding it difficult to correlate. Yes, that is the thought behind this question, sir.
That I understood. I'll tell you, because there is a spectrum of there is a color spectrum or the light spectrum. So there are certain insects which will be getting attracted to the far end, the range. There are certain insects which get attracted to the ultraviolet, and there are certain insects they have the attraction only towards the visual spectrum. So that's why if you I don't know. So whether you have visited the northern part of Karnataka or not. So for containing the Colotrachia serrata and the consanguinea, otherwise what we call it as root grub, and even in case of Insect albistria and Insect mori, that is the hereditary caterpillar in the red gram. So and even the Herpes Polonia and Herpes materna, which is the fruit sucking moth, to contain them. So farmers we actually did not do any sort of the scientific work on that. They used to set the bonfire around the fields, and that is a natural technique they have followed. So in fact, all this is that I have quoted the examples of. They didn't go in search of the fire or the light. They got attracted only towards the color. That is a yellow color, visible spectrum, and that's all. This experimentation in the tritrophic interaction, insect plant interaction, it is there since the dawn of the history, ever since we started cultivating the plants.
Okay, thank you for your inputs. Thank you very much.
Good afternoon, sir. Can I come in for a second?
Of course.
First of all, I would like to extend my thanks for this wonderful information which you have shared with us. Thank you very much.
Thank you, sir. Thank you very much.
Sir, my query is, it's not a question, it's to my understanding also.
Of course, you can please.
Yeah, what is the scope of using pheromone technology in IPM?
Yes. So the pheromone technology in IPM, it is a lot of scope. But in what way you want to use? If the pest is abounding, so the mass trapping technology is there. Like over 40% of the males, if they are caught, that is called a mass trapping technology. And if it is only for the monitoring purpose, in this areas where the negligible pest is there, that is only to ascertain whether it is going to reach the ATL or not. But if all these chemicals are the chemicals are the pheromones that has been talked about in previous sessions also, if they are judiciously used, if they are judiciously used, they of course have the great future. Otherwise, like the PB ropel, so maybe like some 20 years ago, so they thought of bringing it to the market, but the only thing is that there was some hurdles in the way, like it cannot be used in the community basis. They should not. There are several other conditions. So these are all practically possible, but the only thing is that so we have to look at the other side also. Like we always feel like it's always green on the other side, but the other side is not always a green. So there will be certain ups and downs, and the loss will be there. So that also we have to look at before we advent into these things.
Sir, do they have any role in this insect disease management also?
I haven't ever come across any such sort of literature, but in fact, I had 123 slides. Very good question you asked. I'll tell you. One of the slide, some of the information, like the VAM, vesicular mycorrhiza. So this is also not. So once there is the reason as to why this is used. So the mycelial mat, the top of mycelia, which is going to forage or amplifying into the system. So that again is to elicit. It is to trigger a certain sort of defense mechanism by the plant itself against certain insects.
But not my experience. I would like to share my experience here.
Please, sir.
Yeah, one of the pheromone, which is a female sex pheromone, Gopali. You might have heard about that.
Of course.
Gopali. Yeah. We have tried to explore the possibility of using this against the protozoan disease in insects, and we have found some. We have found some encouraging results. But the enough data, as you rightly pointed it out, that enough data is not available to support those findings. So that's why I sought this clarification.
So that is the case. Like you should continue working in that line. It is not my advice. I stand. It is my opinion. It is not my advice. I tell you. So I am not here to advise anyone. It is my stand. You continue in that because what we have seen hitherto is that the insects. Plants will have to have the horizontal resistance against the insects, and the insects, the diseases can be countered or mitigated by using vertical resistance. And if such sort of experiment you have done, and if you have got a very enduring or the very convincing results, so that can be tried or continued rather.
Yeah, we are trying to replicate that.
Please, sir, please.
Thank you very much, sir. Thank you. So nice.
Welcome, sir. Welcome.
Yeah. Good afternoon, sir.
Okay.
Hello. Yeah. Myself, Dr. Swarnali Bhattacharya. I'm speaking from West Bengal.
Madam,
so, it's a wonderful lecture, and really, it was wonderful knowing a lot of new things. So, sir, I have just a query, right? Like, can we use the plant growth promoting rhizobacteria like Bacillus subtilis or so to either maybe it in seed treatment or in soil treatment, so that we can induce resistance against insect herbivory? Is it possible?
Madam, frankly speaking, I haven't ever heard of such things. But we have to look into the literature. Sufficient data should be there, because see, any sort of experiment or any sort of the adventure that we want to do, that should not be a fertile exercise. But of course, before advocating it to the farmer, advising the farmer, what we can do is that we have to have this technical support. But what you have tried, what you have seen, of course, I have not seen. I don't have the experience, so I don't have any comments from my side, please.
Okay. Thank you, sir.
That's okay.
Sir, one clarification.
Please.
So especially in fruits or vegetables or any other crops for that matter, as they are going to produce more of secondary metabolites in response to herbivory. Whether such plants, if you consume those, they're really, nutritionally they are better than the non-infested ones. Any idea in that line?
One wild guess that I have is whenever a fruit is eaten by or tasted or relished by the parrot, they are always good. But the insect, of course, if there is a disease attack, for example, I don't know. So the bananas in the North Karnataka, have you seen?
Yes, sir. Yeah, sure.
There will be 6 spots in the banana leaf. The skin itself.
Okay.
Do you know that is a disease?
Yeah, true.
Anthracnose. So what exactly will happen is that, like, one, there is a anthracnose infection. So the sugar content in the banana is going to, sugar content is not exactly it is increased. So they start degrading into the sugar. That's why they are sweeter. This is the example that I have. But apart from that, I have not tried eating or like consuming any other products that as you stated now.
Any work there where they have quantified the metabolites or phenols or antioxidants in infested fruits or vegetables versus non-infested? Any such studies, sir, as per your right?
At least I have not come across any sort of such
literature. They might have
world. Yeah. Okay. Yeah, I think that is required actually.
In fact, that is required.
Yeah. Thank you.
Welcome, sir. Girish, any question from your side?
So Satyanarayana sir, can you hear me, sir?
Of course.
Yeah, thank you.
Loud and clear.
So your lecture was very informative. You covered very good aspects about insect plant interactions and all. I have one small doubt in this one. You have used the 5 species of this trichogrammatid.
Yes.
Trichogramma, Pidiosum, Japonicum, Braziliensis, Achaea, and Chilonis. That too on cotton ecosystem, maybe under laboratory condition. Do these 5 species, other than this Chilonis, work in cotton ecosystems?
See, the basic thing is that the Trichogramma chilonis, anyway, it is an oak. It is under use, and the literature speaks like Pretiosum and the Braziliensis. The Pretiosum and the Braziliensis, not in India, but the other parts of the world. So the western part, they are using it. They are using. That's why. So whatever the stray reports were there, I tried to. That's why, like, I could have just used the Trichogramma chilonis alone in Indian condition because they are being used. But in one of the experiments, like, I could see even the Pretiosum. So that's why I have tried both the species. So for the finer details.
So how you have managed to rear all the 5 species? Because if any admixtures happens between the species, it was very difficult to separate them.
Very good question. In fact, like, I didn't go for. I know the hardship in rearing the first go, like, the Carceres aplonica and Acor parmigira. I didn't go for rearing all these species. I directly procured from the PDBC. So now it is the NBIR. And there is one more question probably I was expecting from the audience side. So while releasing the mated pair, how did you do the sexual dimorphism? I mean, like, how did you do the sexing? So that is, see, there is one aspect, like the antenna. The second one is the genitalia. Antenna and the genitalia. So we can see the trichogramma is so tiny. Every time you cannot dissect the antenna. Every time you cannot dissect the genitalia. So once you do it, it is only through your experience. Like, so the females will be a little bigger compared to the males. So yes.
No, actually, you told that you have procured these species from PDB, PDB. So there will be a difference in the agroclimatic acclimatization between the Bangalore condition and the Dharwad condition. So with your experience, can you suggest that the biocontrol agent, which is reared and mass produced in anywhere in India, could be used anywhere in other places also?
Okay. So the ambience or the environmental conditions or the abiotic factors will also again have the significant impact on the performance of the bioagents. That is true. So for my lab studies, so for the olfactometer studies, I directly procured it from the PDBC and the TNAU. And once I came to know, like, Trichogramma chilonis is the most appropriate one, we have reared the Trichogramma alone in the laboratory. Otherwise, we didn't go for the exercise of rearing all the 5 species, which is practically impossible. Trichogramma chilonis alone is the one species that I had actually reared in the laboratory.
Thank you, sir.
Welcome.
Dr. Satyanarayana,
please.
This is Gangadhara. Can I add a few things to Harish's question? Harish's first question.
Sure.
Most welcome, sir.
Yes, Harish. It is regarding use of salicylic acid and jasminic acid in pest management.
Yes, sir.
Harish, besides getting good results, effective results with jasminic acid as well as salicylic acid, studies conducted somewhere else and also in our own university. from the Department of Entomology.
Yes, sir.
The concern is jasminic acid cost. It is very high actually.
Yeah.
So between salicylic acid and jasminic acid is very costly.
Yes.
In fact, Dr. Nadaf also have same the feedback. He had conducted used jasminic acid for managing pest on citrus.
Yes, sir.
The cost is the major problem. Otherwise, effectiveness proven beyond the doubt in many cases. So that is the reason we started shifting using of our jasminic acid to the salicylic acid. Salicylic acid is relatively, the cost is less as compared to jasminic acid.
That's only 600 rupees a kilo, sir.
Yes, and in fact, chitogen also, as you mentioned, that also we started using in many cases.
Okay.
That's the one thing. And regarding Bhattacharya madam, Satyanarayana?
Yes, please.
Madam asked about using of PGPR in pest management. Again, we have our own experience in the case of onion thrips. Where we use Pseudomonas and Bacillus subtilis. In fact, they given comparatively better results as compared to the minus PGPR. And even we went for the biochemical analysis also. So what could be the reasons for the lesser pest population, thrip population recorded onion, where we have used the Pseudomonas as well as Bacillus subtilis. We mixed in the soil. The reason was wherever you used Bacillus as well as Pseudomonas, the phytochemicals like the phenol as well as tannin content were high, significantly high in the case of PGPR used the plants as compared to the non-PGPR used plants. So that given an indication, probably wherever you use Pseudomonas, fluorescence as well as the Bacillus subtilis and such kind of a PGPR, probably not to higher extent, some extent it induces the production of the phenols as well as the tannins. That is our first understanding. In fact, it was the study conducted by the MSc student, and we are looking forward actually to confirm in a systematic way. This is first sort of response we got from using up PGPR for managing at least a second pest, thrips in the case of onion.
Thank you, sir.
Yes, Gangadhar sir.
So if
you don't mind, I have a query in this regard because you have answered that one. Have you done the complete chemical profile of the extract? It's only the phenols, and you said one more thing. So
phenols, tannins as well as ascorbic acid.
Okay.
Only three we could analyze in the case of biochemical analysis.
Probably the standard check you have taken, and there is a variation.
Exactly.
That is good.
Yeah.
Sir, I would like to add one thing because, sir, I have done a little bit of work on this also. So, peroxidase level also is shown to increase actually, sir. So, all are linked with the defense mechanism of the insects, basically.
Exactly.
Okay.
Thank you, madam. Thank you for your information.
Thank you, sir.
Okay. Any questions from the audience side, please? Otherwise, I will try to check the queries if there are any in the chat box. Sir, there is a question like, sir, any roles of push component technology for the management of rodents? So there is only a baiting technology which we know. So push technology, if there are, I really doubt if there are any push kind of technologies. So the pull is there anyway, and the push I really doubt. And there is one more question. Hello, I am Dr. K. Okay. There is only one question from the audience side of the chat box. Okay. Any more questions from the audience side, please? If there are no more questions, what we'll do is that we proceed to the valedictory session. Any questions, please? Thank you, sir. Thank you very much. Thank you, all the participants for having been very patient till the end, and there was a nice interaction at the end also. In fact, there are people who came for in supporting the queries, so which I was totally into the dark. Gangadhar, Girish, and Harish sir, thank you very much for your the active interaction and all the participants. Of course, the one more madam from the West Bengal also. She interacted nicely with the Gangadhar Narvinchi sir. And Gopali sir, are you there?
Hello, Satyanarayana.
You are there. You are logged in.
Yes, I am already.
One second. Okay, please.
Thank you very much, Dr. Satyanarayana C. So, being an organizing secretary, so at the outset, so has given his lecture on this recent advances in insect plantation infections for the pest management. So, it has got to be very much useful information for all of us, and so we could able to gather some of the information through interactions with other scientists as well. So, we professionally convey. Thank you for presenting a very useful information in this platform and making graciously this particular explanation. So, thank you, Dr. Satyanarayana sir, for giving this
Thank you very much. Thank you, Srinivas. Thank you very much. Thank you, Dr. Srinivas. ಇವರು ಡಿ ಸರ್ ಇದ್ದಾರೆ, ಡಿ ಸರ್ ಲೈನಲ್ಲಿ ಇದ್ದಾರೆ, ಇವರಷ್ಟೇ ಲಾಗಿನ್ ಆಗಬೇಕು. ಡಿ ಸರ್ ಇದ್ದಾರೆ ಲೈನ್ ಆಗಿದೆ. ನಮ್ಮ Horticulture ಸರ್ ಇದ್ದಾರೆ. Gopali sir, we are expecting Dean sir to log in.
Okay, no problem.
Dean sir, Dean sir, are you there?
Very much there.
Yes. Okay, sir, please. I'm expecting our Dean, Ravindra sir, also to log in, please.
I am there.
Okay, sir. Thank you, sir. Thank you very much. First and foremost, I am thankful to all the patrons and the organizing committee. So we have been very supportive for this particular program. And first and foremost, I would request I welcome the Dr. YK Kotikal sir, Honorable Director of Extension, UHS Bagalkot, University of Horticulture Sciences, Bagalkot. And I also welcome our Dean, the President of the Valedictory session, Dr. Ravindra Mulge sir, Dean, College of Horticulture, Bidar. Welcome to the program, sir. The Kotikal sir and the Ravindra Mulge sir, welcome to the program. At the same time, I welcome the Please, sir. So I welcome the Dr. J.V. Gopali, Professor of Entomology, RHRS, Dharwad, affiliated to the University of Horticulture Sciences, Bagalkot. Welcome to the program, sir. Gopali, sir, welcome to the program.
Thank you.
Okay. So the first and foremost, I would request the chief guest, Dr. YK Kotikal, the Honorable Director of Extension, UHS Bagalkot, India, to expostulate your opinion about the training program, sir, please. So I know that you have a very busy schedule, sir, but still and we have troubled you a lot to give your time. Please express your views, sir.
Okay. Good afternoon, everybody. Respected President of this concluding session, Dr. Mulge, my colleagues, Dr. Dhaneshwar Gopali, the organizing scientists, Dr. Satyanarayana and others. Of course, it was a good seminar. I was just listening to the speech very recently held by Dr. Satyanarayana. and there was a very good discussion also taking place. I was very much impressed by the way you have been participating and interacting with each other. All the lectures which have been selected for this seminar were very good, and the resource persons have given a mature talk. And I came to know about the list of the participants also. Very good response was there. I've been seeing in all the 10 good participants, and they have taken a lot of things to from this seminar. They also contributed, and there was a mutual benefit for the participants. Since I was also not that free to listen to all the lectures given by scientists during all these 10 days of this seminar, few of them attended. It's good. I'm very much impressed by the way it was been conducted timely. It's good, and I wish all the participants good deliberations and good thoughts to be taken away back home from this seminar. Thank you, and I give the I thank the organizers for giving me this opportunity to give this conclusion. Thank you.
Most welcome, sir. Most welcome, sir. In fact, we are very thankful to you because I know that you had a very busy schedule, and even now also you are on wheels, but still then you cut out a time for us to spend some time, and you attended, and that's an honor for the entire program, sir. Thank you, sir. Thank you very much.
Thank you.
Okay. Now I would request our Professor Dr. J.B. Gopali, sir, to continue with your valedictory remarks, sir. Please, sir.
Okay, good afternoon to everyone participating in this webinar. Today we have to come to an end to the webinar, and happy to be with you all on the occasion of this valedictory function. First of all, the chief guest on the occasion, Dr. Y.K. Kotikal, Director of Extension, University of Horticultural Sciences, Bagalkot. And secondly, the President of the function and Dean of COS Bidar, Dr. Ravindra Mulge, the man who is instrumental and deserved in conducting this online training program successfully. And also the Dr. Satyanarayana, who is the organizing secretary, who is the pillar of this training program, and supported by this pillar by the many joint secretaries, Dr. Ganesh Gowda and Raman Gaud, and all the staff of the COS Bidar, and all the distinguished members of the secretaries, and all the entomologists who have involved directly and indirectly in making this training program successful. Indeed, it is a great pleasure for me to share the valedictory remarks of this 10 days national level online training program entitled Recent Advances in Entomology, New Dimensions to Invigorate the Insect Pest Management, which started in fact in this month from 7th December to till today, that is 18th December 2020. So I am happy to note that this webinar in the beginning, it was inaugurated by our Honorable Vice Chancellor Dr. K.M. Indresh as a chief guest on the auspicious day of 7th December 2020. And I recollected his remarks that made by our Vice Chancellor that so first of all we have to use the safer methods of controlling the insect pests and with no toxic residues on the food commodities. So that has to be given the main priority as remarked by our Honorable Vice Chancellor. And similarly, the guest of honor chaired by Registrar, University of Horticultural Sciences, Bagalkot, insisted the entomologist to create more and more awareness on biological control, natural way of control, organic farming, which are being accepted by many of our farmers. And that is the only way for sustainable management of plant protection. And he also underlined the need to develop the pheromone traps and different kinds of traps for management of the various pests so as to reduce the usage of the toxic chemicals. Further, our beloved Director of Research, Dr. D.R. Patil, University of Horticultural Sciences, Bagalkot, also called for efforts by our scientists to address the various issues of the pest management. Similarly, another dignitary, Dr. Y.K. Kotikal, a renowned entomologist and also the director of extension, University of Horticultural Sciences, Bagalkot, he also advocated the implementation of IPM, which is most important, and also the use of latest technology in pest management, use of new molecules with the integration of bioagents, green-labeled molecules, which are eco-friendly and economically viable and easily adaptable by the vast majority of the farmers. And he also trusted the time of application of either bioagents or insecticides based on ETL, which assumes a greatest importance than just mere application of improper way of application of the insecticides and also the timings. Yet another multi-personality, Dr. Ravindra Mulge, in COH Bidar, on his presidential address, he stressed about the need to breed the crops for insect resistance, which is the key component and integral part of the IPM. So he also stressed more on the host plant resistance, HPR. So that is also a key component of IPM. And the keynote address delivered by Dr. Venkateshlu, HOD, Department of Entomology, COH Bagalkot, he also emphasized a new concept called push and pull technology. So we have to push the insect and we have to pull the natural enemies, like that, attract the natural enemies. So that is very simple technology, which was thought-provoking and informative as far as pest management is concerned. And for successful completion of any task, a teamwork is important, and that plays an important role. So this Herculean task is not possible by one hand. So it needs a strength. So in this regard, role of organizing this secretary by Dr. Satyanarayana, who is known for his leadership quality and strength in organizing this training program, and his team also committed and totally devoted for successful completion of this training program. Coming to the speakers, as far as the speakers is concerned, on the first day of the opening, the first batsman was Dr. Professor of Entomology, US Raichur. So who was invited in the afternoon for the presentation from 2 o'clock to till 5:30, and his topic was ESAP, an ICT solution for crop health management. So he stressed ICT. Nowadays, as of all of us, we know that the Android mobiles are everybody. So now, because of this invention of this Android mobile technology, so all the technologies fingertip. So if you just click using the finger, everything we get. That is the ICT invention. So this is the novel tool used in agricultural and horticultural system, and it can be easily used by the extension functionaries as a diagnostic tool and also as an advisory expert. So it is very useful for administrators, researchers, policymakers, and other stakeholders for the management of insect pests and diseases, and also the nutritional disorders due to various nutrients. So he also stressed that this ICT technique operates both offline as well as online with the built-in robust information on pest and disease, as far as at what time the pest will appear, identification of the pest, and the right time application of the insecticides. So all those things are integrated in this ICT tool. And another important feature of this ICT ESAP is that it operates on real-time basis. The farmer can obtain the information at any sitting at any corner of the universe. So farmer can get the information on online, means as and when he wishes, he get the information, and he can send the information directly, even in the form of photographs, videographs. Sitting in the field, he can send the information to the scientist, and in turn he will get the solution immediately for that particular problem. So that's how the importance of this technology is very much useful in nowadays pest management. And this ESAP also helps in forewarning and forecasting of the pest and disease. It is most important. as far as pest management is concerned. So this is all about, he elaborated in detail about the so many things, but here just I highlighted his, the gist of his summary of his topic. On the second day of our presentation by Dr. S. Subramanian, IIRI New Delhi, who nicely presented his topic on characterization of gut microbes, that is the endosymbionts. He explained in detail about the importance and structural functional characterization of symbionts. For example, sucking pests and chewing pests have different gut anatomy as they feed on different types of foods. So these morphological techniques that will help in gut microbes, they have the significant impact on insects, and for that he has explained different techniques like cultural dependent and cultural independent molecular techniques and rDNA primer, next generation sequencing, so many things, so many the DNA level information he has given. And he also explained the factors influencing the colonization of the microbes, which are pH dependent, oxygen dependent, and in anaerobic absence of the oxygen and redox potential. So many things he has explained. And apart from that, gut microbes also plays an important role as far as the detoxification of the toxins is concerned. So, for example, you might be knowing that the Helicoverpa arimigera, when it ingests any toxic material, it immediately expel or excrete so as to survive itself. So that's what in this regard, the detoxification of the ingested toxins. So the gut microbes also plays an important. And similarly, the different types of bacteria associated with the insects, they play a significant role, and they play a symbiosis role as far as insect is concerned. And they also provide the nutrition. So that's why the nutritional symbionts, cellulose decomposing or cellulose digesting enzymes, are provided by the microbes, which is most important. Otherwise, the cellulose cannot be digested so easily by the insect because of the insect, because of the the microbes that are exist in the gut that helps the insect to digest. Like that, even another aspect also, insect plant interactions and insect natural enemies interactions and resistance in insects. So plant defense mechanisms. So many relevant things he has explained. And on the third day, Dr. Ankita Gupta, senior scientist, NBAIR, Bangalore, she has made a very successful attempt to draw the attention of all the participants towards the importance of integrated taxonomy and its role in agriculture and diversity studies in pest management in particular and in agriculture in general. So she explained the biodiversity studies and also the species extinction. And she explained the exploration of the specimens for passive collection through traps, and also she also explained about the methods of species of diversity estimation using her decades of knowledge. And also she explained the global pattern in braconids and Cotacea species diversity, transcontinental level of diversity. So especially the Cotacea species was identified just by the way of egg-laying pattern. This is one of the instances. Like that, Ankita also elaborated about the experience working at a place where there was active volcano. And like that, she has also explained the morphological, biological, molecular analysis of the so many genuses in insects and host range, the genetic boundaries, and also the biodiversity studies dealing with the tritrophic interaction, and taking the example of vast biodiversity, and also the host range expansion from the Papilio-Demolis, that's a citrus butterfly. And like that, she has explained us many things regarding the taxonomy. And another scientist on the fourth day from NBAIR Bangalore, that is Dr. Richa Vasni, who also stolen the attention of all the participants towards the importance of vibrant topic on the status of augmentative biological control in India, challenges and opportunities. So she explained so many things, different methods of biological control, how to conserve, and classical examples, and how to augmentative release of these biological agents, taking the examples of trichogramma, various species of the trichogramma, and also Telonimus, Goniaceus, and Braconids. So many predator rats. And she also explained the new potential predators like anthocorid bugs and mirid bugs, geocoris species. So many things she explained with case studies. So 2 Encacia species used against a coconut spiraling whitefly. So that's which is one of the most important case studies she explained. And biointensive IPM in rice, trichogramma and Pseudomonas, which are the important things, which covered 75,000 hectares of area. This biointensive IPM. And recently this false armyworm, which can be controlled by a natural enemy, that is a Telonimus remus and Trichogramma chelonis. And another predator mite. So is used against the red spider mite. So like that, she explained even the ASRP and biological control, covering the coconut rogose whitefly management, sugarcane woolly aphid, and so many things she explained. And apart from that, the commercial insectaries established in Angrul and US Raichur at the NBAIR also explained. So she explained many things on the commercial establishment of the biological control and so many other things. And on the fifth day, Dr. Ravi Kumar Hosmani, Assistant Professor US Dharwad, about the development of nanopesticides for crop pest management, which is most relevant for today's pest management is concerned. And he covered in detail history of the nanotechnology. It is a very exhaustive about the nanoparticle, how to increase the efficiency and also use of this nanotechnology. So many other things related to this nanotechnology formulations, nano capsules, nano gels, nano emulsions, so many things he explained. And apart from that, nanopesticides have slow in action, hence they can only be used as prophylactic, not in the pest outbreak. Of course, pest outbreak, even once the insect gets outbreak, even the insecticides cannot help. So leave about the biological agents. So that's why these nanopesticides have low, slow action, and they have to be used as a prophylactic, as a precautionary measures. And also he explained the experiments conducted on prophenophos technique against the Spodoptera litera, and also the bioassay, different types of bioassays, how to assay the bioassay of different insecticides against different pests using leaf dip method and lipid encapsulated water and lipid encapsulated, all those things. And apart from that, he also explained the green synthesis of metal for pest management, metal nanoparticles for the pest management, that's EGNO3. So compared to gold, copper and other metals, silver is mostly preferred as far as the formulation of the nanoparticles is concerned. And also he explained the characterization of green synthesized silver nanoparticles in detail. And still he stressed a lot of work is required as far as the refinement of this nanoparticles is concerned. And further, myself covered on recent trends in vegetable pest management, where the importance of the vegetables in day-to-day's the health management is concerned, how the vegetables plays an important role in maintaining the health and also in curing the diseases, how the vegetables they are helpful. And also the reduction in the vegetable yield, the factors that are responsible. So many factors are responsible for that in the reduction of the vegetable production. Biotic and abiotic factors are responsible, but among those, the insects plays an important role in reducing the production and productivity of vegetables. And one thing we should remember that any intervention in the environment towards the elimination of a species brings out parallel change in the pest to overcome the hurdle. This principle was overlooked till recently, but now it is time to rethink, relook and reconsider this principle. So we should not try any species the elimination, towards the elimination. Elimination is impossible. So we have to manage the, somehow we have to manage the insect. And because even 100 years ago, our farmers, they were not using any pesticides and chemical fertilizers, but now it becomes common practice to use. Without the insecticides, it's impossible to cultivate. To that stage we have reached. So because of so many reasons, maybe the monocropping of the vegetables or the indiscriminate use of insecticides, especially the conventional insecticides, organochlorines, organophosphates.
Dr. Gopali has lost the connection, it seems, net problem.
That's a problem with the bandwidth, sir, actually. That's a
Just
problem.
call him and confirm.
Yes, sir. Yes, sir.
Dr. Ganesh, Satyanarayana,
Yeah, I'm just calling.
just call him and inform him that he has lost the connection, net problem with sand.
ಗಣೇಶ್ ನೆಟ್ ವೈಸ್ ಕಾಣ್ತದ ಗೋಪಾಲ್ ಸರ್ ಏನು ಮಾಡೋಣ ಸರಿಗೆ ಮಾತಾಡಕ್ಕೆ ಸರ್
sorry, dignitaries and all the participants, there is a technical snag, there is a bandwidth problem, it seems from the other side, from the Dr. Gopali sir. So now what I would do is the Dr. Ravindra Mulge, Dean Horticulture, College of Horticulture, Bidar, who has been who has been the backbone of the entire the program. He stood by with us, he stood by us when we are going through thick and thin. And without his support, all the comments and all the comments that we are receiving, actually, in fact, in the chat box, it's only the credit goes to Dr. Ravindra Mulge sir, who has been the
the rock solid support for the entire program. Sir, the Gopali sir has actually lost the connection. I mean, like it's a bandwidth problem or the net drop problem. I really don't know. So what I would do is that, like, I would request your kind sir to give the presentation remarks and will address the program, sir. Welcome, sir. Welcome to the program again.
Okay, thank you, Dr. Satyanarayana. Yes, I think Dr. Gopali has almost reviewed the entire training program. I think maybe I left with a few more points. Hope he has completed almost everything, whatever has been covered in this training program, right. Respected Director of Extension, Dr. Y.K. Kotikal, Professor Dr. Gnaneshwar Gopali, Dr. Satyanarayana, and all the participants, and maybe some of the guests also participated in the valedictory function. I really thankful for our Director of Extension for making himself present here for the valedictory function and giving the remarks. Thank you, because he was very busy. This Horticulture Mela is being planned in the first week of January, so has very little time for that and a lot of other engagements. Dr. Gnaneshwar Gopali, you are back. You would like to conclude by saying few more words, so then again I can start.
Another 5 minutes, sir.
Yeah. You go ahead. You just start and you finish it.
Network is the bandwidth is a little bit low in our station, so that's why there was some interruption in between. So I will continue with this one.
Please.
And another thing is that there is one new technology that is called as an ecological engineering. Ecological engineering means it is a recently introduced or emerged a technology that is considered for pest management practices based on the cultural practices, especially based on the cultural practices coupled with ecological knowledge, which is most important. Otherwise, what the farmers are doing now with a unilateral approach such as synthetic insecticides, they are managing the pests. But we have to consider ecology also. Ecology is most important, both for the pest as well as the natural enemy. So if we know the ecology, knowledge on ecology, it is easy for us to manipulate the insect behavior and also the insect habitat, making less favorable for the pest and more attractive to the beneficial insects. That's what our HOD explained, the push and pull technology. So this helps a lot, this ecology. Knowledge on ecology, the goal of this ecological engineering is to implement suitable crop diversification. Crop diversification means that we have to promote the biological services. Crop diversification is must, such as growing intercrop, cover crops, the trap crops, and also the so many other crops that makes the crop diversification. Because this crop diversification in future, it becomes an important integral tool of IPM. As I was telling, it includes the integration of barrier crop, border crop, intercrops, along with the main crop. This is the concept of the ecological engineering that provides so many principle or concept behind it, as a physical barrier to prevent the movement of insects and so many other things. And this helps that for natural enemy, it attracts the natural enemies and it repels the pest. So that is the diversification. Diversification is most important for implementation of these two components. That is to repel the insects and to attract the natural enemies. That's what our HOD told, that this is a push and pull technology. Push and pull technology. So we have to pull the natural enemies and we have to push the insect. So this is crop diversification. Crop diversification plays an important role. And apart from this, so many other IPM module. That is the crop diversification based IPM modules were explained on major vegetables. And on 7th day, another scientist from IARI New Delhi, Dr. Sagar, who presented his topic on impact of thermal stress on physiology of the insect. So he made so many definitions on his topic, the stress factors related to this, his topic. And he also explained the radicals, that is the hydrogen peroxide and hydroxyl ions. That's how they impact the thermal stress on physiology of the insects. So this topic is a little bit difficult to understand. It is possible only if we understand the physiology of the insect. So he also mentioned so many defense mechanisms in insects against the stress, like antioxidants, heat shock protein, anti-stress hormone, cuticular hydrocarbons development, and cryptoprotectants. So many things he explained that impact the thermal stress on physiology of the insect. And on the 7th day itself, another scientist, Dr. Suresh Nabapur, also presented his topic on insect semiochemicals, which are most important as far as crop pest management is concerned. He covered in detail about the insect-plant interaction through these chemicals, semiochemicals, and the importance of these chemicals, role of especially the role of plant volatiles, how they help in attracting the natural enemies and also repelling the enemy pests. All those things he covered. And he also covered the pheromones monitoring, mass trapping system, and use of different lures to kill the insect, mating disruptions. So many things he explained. And on the 8th day, Dr. A.G. Srinivas, Professor of Entomology, US Raichur, he also explained the impact of climate change on transgenic crops through insect pests and their management. The climate change is nowadays playing an important role, and everybody is talking much about the impact of climate on human beings. But there is also impact on insects, whether there is a multiplication, whether there is an increase in the pest population or decrease in the pest population. That is most important. So as there is an increase in the carbon dioxide content in the atmosphere, there is every chance of development of the heat. For some of the insect, it becomes a positive, and some of the insect, it becomes a negative. So all these, the studies have been dealt in detail by Dr. A.G. Srinivas, and he also explained the changes in the plant architect. That's how the changes in the biochemistry of the plants, making them more prone to insect attack. And so because of this, the impact of climate change, there is a change in the ratio of the oxygen and carbon dioxide in the environment. And if there is a change in the oxygen and carbon dioxide content in the environment, there is also change, there is also alteration in the CN ratio, either both in the plants as well as in the insects. So that has the impact on the growth and the development of the insects. So that's how he explained many things. And on the 9th day, Dr. Venkateshlu, Professor of Entomology, and also he is the head of the Entomology Department of Entomology, RHRS Bagalkot. He also presented very nicely on a topic called as application of push and pull strategies in pest management. So he explained very nicely the basic principle and concepts of push and pull technology, and what is the meaning of push and what is the meaning of pull. So that is what this strategy used for diverting the insects, push and pull technology. So pushing means what? That's what it's called as. Pushing means repelling the insects from the main crop. Repelling the insects from the main crop is the push. Pull means that attracting insects towards the predetermined crops. That's what trap crops or border crops or intercrops, like that. So he explained very nicely this principle, and he also explained the population reduction methods like using the microbial control, bioagents, mass trapping devices, and also the dead and attractive crops. That's also most important. And also he explained why we should use this push and pull strategy. He explained very nicely about this technology, and with examples, he explained the behavioral parameters of the insect. In India, where it is used, he also explained, especially the only trap concept is used of late. Push and pull strategy is being introduced in African tall marigold. That's one row of African tall marigold. And mustard, in cabbage, like that. It is using this technology. Like that in pomegranate also, how to ward off the fruit sucking moth. For this, push components are neem oil and lemongrass oil and citronella oil. They act as a push components. Pull components, brinjal, banana, apple, citrus as intercrops. So that's how push and pull technology plays an important role as an integral part of the IPM march. And on the last day, that is today, our organizing secretary, Dr. Satyanarayana, he explained the recent advances in plant interaction for pest management. Very nicely, he explained the various reasons for the explorative studies on the biocontrol as far as management of the insect is concerned, and also various tritrophic relations and different dimensions of insect-plant interactions with respect to morphological types and the using alkaloids, antioxidants, and so many things. And also he explained the exploitation of the intricate relationship among the different food and trophic levels for the pest management. Like that, he many participants appreciated his way of presentation, and also the coverage of the subject especially is really appreciable. So with these remarks, sir, I finish my valedictory speech, and I thank one and all for this successful completion of this the 10 days national level training program. Thank you one and all.
Thank you very much, Dr. Gopali sir. It was more than a valedictory ceremony. It was like you have paid so much of attention to the entire proceedings, and for having put everything very concisely and systematically in a nutshell. So that takes a lot of time and the patience also. I am thankful to you, sir. Thank you very much, Gopali sir. Now I would request our beloved the Dean sir, the Dr. Ravindra Mulge sir, so to express few words through your the presidential address, sir.
Thank you, Dr. Satyanarayana. Yeah, I just started giving my remarks. Thank the Director of Extension, Dr. Y.K. Kotikal. And yes, all the entire 10 days program in just half an hour, that is really appreciable. So everybody would have got the insight of what has been done from the last 10 days. That's really great. Yes, inaugural session, it was by our Honorable Vice Chancellor and also Registrar. Really, we are thankful to them for giving us all the opportunities and also giving the permission for carrying out this important training program. I'm really thankful for them. This program is really started with a good intention. But I just remarked at the beginning also, inaugural session also, doubling the farmers' income, where almost 40% of the food produced in the world is being spoiled, and 20% is being contributed by the insects. So that speaks about huge that loss. If you can really curtail it, you're really helping the farmers to get more income. That is one way it is very much attached. So management of the insect pests by sole use of chemicals, you are looking for the other opportunity. That's really a good intention of this training program, which has been covered. So many topics are just like that, maybe what advances you would like to have in this particular field. It is ESAP and ICT solution for crop health management. That is the efficiency of the entire plant protection measures. It speaks about how you can take the present days developments in the field of ICT. That's really commendable, and one should go for it. And also status of argumentative biological control, its challenges and opportunities. It has been covered. Really, it is away from use of chemicals. We are always looking at it. It doesn't mean that we should not use the chemicals, but at least we should reduce it. But we should reduce it with the view of integrated pest management. And of course, the development of nano pesticides for crop pest management. It's a new development of how it is going to be useful and what are the advantages and disadvantages of this approach in managing the pest. It has been dealt and really appreciable. And thermal stress on physiology of insect. This is very important. Why I felt is if you know the physiology of the insects by, how the temperature is influencing, can you find the solutions for the climatic changes, right? We are going to face in near future. I think our approaches should be different. It's not only just, some biological control or some other method. Just if you know, can you, by changing the temperatures, how the different group of insects, their multiplication rate, their life cycle is going to be influenced. So that really can give you a solution for this big problem. And yes, genetically engineered crops, yes, that is, for breeding. So plant breeding is really not very successful in breeding against the insect pest. If you compare this with the disease resistance. Yes, disease resistance, we have made a lot of developments, but for the pest resistance, insect resistance, very little has been done because these insects are more evolved and they are near to the human species, and it's difficult. Something you get the chemical like, tridecaron, you say it in tomato related species. People used to say. So such toxic chemicals are also toxic to the human being if you have your variety. And even today also by genetic engineering, this BT approach, still people have the apprehensions regarding the technology because how this is going to be safe for human being. That's why crop breeding or genetic engineering is still a second option for this breeding, the solution to the pest compared to the diseases. So that's why the approaches what has been looked at, cultural and ecological methods, really should help reducing the cost, knowing that push-pull technology, what has been dealt on. This is crop diversification or trap crops. These are the very finer issues one should look at, and one should find the solutions through these avenues that can really help in reducing the cost and also effective management of the pests on the crops. So I'm very happy that the coverage, whatever it has been done from the last 10 days, some trainees also remarked. Normally these trainings are being organized by some senior professors, but yes, it is commendable that Dr. Satyanarayana, just assistant professor, he could able to organize this training so successfully and effectively. Now, how I am saying I was just reading some of the remarks by these trainees. Somebody said it's excellent training program, wonderful training program. All these adjectives have been added. Knowledgeable training program, really good. That means they are satisfied. Some latest developments has been covered and it has been well discussed. I feel, and that's why it is really appreciable. And someone said it is informative and useful for formulating innovative research programs in future. It's really one of the best objective or outcome of this particular training program. It is really commendable. And someone said it's considered giving opportunity for farmers to participate in such trainings. And I think here, yes, there is no problem. It is not that you are calling, there is no expenditure. It is the corona situation has forced all of us, and this corona only is responsible for having a training program hosted from College of Horticulture, Bidar. Otherwise, it was very difficult. It was very difficult to make all the other arrangements. So farmers inviting in this training program, not an issue, but only thing, language can be a barrier for people to understand that. But yes, you can get the feedback from the farmers. So all these issues have been really dealt, and which has been elaborated and coverage of all the topics by Dr. Gopali. So this training, I felt this is really very fruitful, useful, and it is not just some certificate, but it is enriching the knowledge, sharing the knowledge among the scientists in this particular field. And I'm really thankful to Dr. Satyanarayana and his team, supported by his professor and head, Dr. Venkateshwarlu, and his department colleague, Dr. Rajkumar, and of course his team like Dr. Ganesh Gaud, Dr. Srinivas, Dr. Mohammad Farooq, Dr. Arun Kumar, and Raman Gowda. So many people have really helped him, but yes, it is his initiative, his effort made this training program more successful and being appreciated by most of the trainees. So I thank one and all. I thank the participants. I thank the whole university, Department of Entomology, really helping our entomology department to make this training program very meaningful, very informative, which is going to be useful for formulating your research programs or even in teaching programs also. Not only research, it is in teaching program also, it is going to be useful. So I'm very thankful to all the people who are involved in this completion of this training program for the entire 10 days. Thank you one and all.
Thank you, Dean sir. It was more than a presidential remark. It was the sort of the encouragement, the moral support, and all sorts of support, full-on support. In fact, it was given by you. Otherwise, we could not have been able to culminate this program. And first and foremost, I would like to thank the Honorable Vice Chancellor, Dr. K. Ravindra sir, and Dr. T.V. Already sir, the Honorable Registrar, UHS Bagalkot, and Dr. Venkatesh sir, Professor at the Department of Entomology, UHS Bagalkot, and Dr. Ravindra Mulge sir, the Honorable Dean, College of Horticulture, Bidar, for having given the scintillating start to flagging of the inaugural session, and all the speakers who took us through the insect world for all these 10 days. And I'm also thankful to all the members who have participated, and I'm also thankful to the chief guest of today's program, Honorable the Director of Extension, Dr. Y.K. Kotikal sir, for the valuable time that you have given. And I'm also thankful to Dr. J.B. Gopali sir, Professor of Agriculture, Entomology, RHRS, Dharwad, for having given your time and the effort that you have made for the valedictory remarks. And I'm last but not the least, all the participants who have been the beneficiaries, in fact, have been very patient listening to the lectures all through these 10 days. And thankful to all of you, one and all. Thank you very much. And the one more announcement that I want to make is, like, let us heave a sigh of relief, because we want to take a day off, and then we'll issue the certificates. And this is only subject to the condition that you submit the feedback form. Thank you all. Thank you very much. Have a nice day.
Thank you, sir. Congratulations. Congratulations.